it looks like we're going to just walk through sort of how I looked at the dual targeting of two proteins, one being APE1, which is a DNA repair protein and a redox coactivator protein, and STAT3, which is a transcription factor. We used a few different ways to do that. Genetic approaches using overexpression and knockdown, as well as pharmacologic approaches using selective small molecule inhibitors of both STAT3 and APE1. We look at the effect on cancer cell survival and migration using the dual targeting approach, and that data that I'll show today is all in pancreatic cancer. And then we also can interrogate tumor stroma interactions using conditioned media and co-culture experiments. So synthetic lethality is the pairing of two hits that is sufficient to kill cancer cells, where either one alone might not be able to do that. And so pictured on the left panel is a non-tumor cell. Those have all the DNA repair protein and pathways intact, and you have live, happily growing cells. Sometimes in tumor cells, they will become deficient in a repair pathway, such as BRCA1 and 2 deficiency. However, this can actually give the tumor cells somewhat of a growth advantage, as they can just go ahead and replicate through some DNA damage. So these tumor cells are able to live, other DNA repair pathways compensate, and the cells are happily growing. The idea of synthetic lethality is that you would take a BRCA deficient cell, or a cell that's deficient in one DNA repair pathway, and hit it with another inhibitor, perhaps in another repair pathway, or perhaps a cell cycle checkpoint protein, some other inhibitor, that by using both of these inhibitors, you then lose two DNA repair pathways in this instance, and then the tumor cells would die. So this is the idea that you're going to interfere with two important pathways where either pathway would not have been sufficient to kill the tumor cell. Now this slide is just to tell you a little bit about the protein that we study in my lab. It's called APE1 or REF1, and it has two names because it was first discovered as a DNA repair protein, which is the APE1, AP endonuclease 1, and it functions in the base excision repair pathway. REF1 is the function that we're going to focus on today, and that stands for redox factor 1, and that is involved in the redox control of transcription factors, as you can see on this slide. So in the left panel shown, the transcription factor is oxidized, and there is very little binding to the DNA, and thereby not activating its target gene expression. After APE reduces the transcription factor, it can then bind to the DNA, activate target genes, and you'll see increases in protein expression. Some transcription factors that have been shown to be under redox control by REF1 are HIF1 alpha, NF kappa B, APE1, P53, and I'll show you some data today about STAT3. So we began this project in collaboration with a couple of labs here at IU, including Angelo Cardoso and Mark Kelley, as well as Patrick Gunning from the University of Toronto. And basically there were a couple of reports that hinted towards STAT3 and APE1 being important and interacting, as well as perhaps APE regulating STAT3's DNA binding activity. First, in pancreatic cancer cells, they found APE and HIF1 alpha and STAT3 all on the VEGF promoter under hypoxia. Then a paper came out in HepG2 cells showing that IL-6-induced STAT3 activity was stimulated when APE-REF1 was present. So our question became, we know that STAT3 DNA binding is redox sensitive. Could APE-REF1 be important in that activation of STAT3 through redox? We are going to use a few different STAT3 inhibitors. The ones that are commercially available are called Static and S3I201. And then from the University of Toronto through Dr. Gunning's lab, we were able to acquire some even more potent compounds that we call BP1-102 and SF1066. So as you can see in this cartoon picture here, STAT3 is activated by through the JAK-STAT signaling pathway. It's phosphorylated and dimerizes, and then it moves to the nucleus. But there actually seems to be one more step of activation in that the DNA binding is then also redox sensitive. So if STAT3 is reduced, it binds more tightly to DNA and can activate transcription more effectively. Just to give you a little bit of background on STAT3 and why we believe it's important, it's been shown in many kinds of cancers to activate genes that are important in proliferation, migration, angiogenesis, and all of these kinds of genes can then lead to metastasis. And in pancreatic cancer, most patients present with metastatic disease. And here's another cartoon showing sort of our hypothesis is we know that our tumors express APE and the tumors express activated STAT3. We also know that there is expression of these proteins in the tumor microenvironment. So shown here is just a cartoon of some stromal fibroblasts as well as extracellular matrix proteins, and as well as there would be some pancreatic stellate cells are important in the pancreas microenvironment, and these kinds of proteins are important because there is this fibrosis that occurs in the disease. And it looks like APE is expressed in both the tumor, as you can see in the brown staining here, as well as in the stroma of the patient tumors. So we want to be able to block APE in the tumor and block STAT3 in the tumor and see what happens to tumor proliferation as well as metastasis, as well as blocking those things in the cells that are supporting that tumor, such as fibroblasts and stellate cells. Just to give you an idea of why this is important, pancreatic cancer is a very deadly disease, as shown here. Most people present with not unresectable disease, and most of them present with metastatic disease. Gemcitabine is the standard of care, and there's been some new therapies that have come out lately, such as the FOLFIRINOX, that seems to be a little bit better than gemcitabine. But in general, there are really no treatment options for this disease, especially when you present with unresectable. Okay, so here's just a reminder of the different transcription factors that APE regulates, and STAT3 is the newest one that we've added to the list, and I'm going to show you some of that data today. So here is some of the staining, looking at pancreatic cancer patients from University Hospital here at IU, and you can see the large arrows point to the APE staining in the tumor cells, and the small arrow point to the APE staining in the stroma on that top panel. The pictures here are phosphorylated STAT3, and you can see the brown again. It shows that there is activated STAT3 in these tumors. These are actually patient tumors that were put into immunodeficient mice. So they're human pancreatic patient tumors that were taken from mice. Panel B just shows westerns of APE protein in tumor and stroma, as well as the activated phosphorylated form of STAT3, and then the total STAT3 that we see in these different pancreatic cancer cell lines. The PANC1005 and PA03C are two low-passage patient-derived lines that Anirban Matra from Johns Hopkins University has given to me, and you'll see some data in those lines as well. PANC1 and PACA2 are two common pancreatic cancer cell lines that have been used for many years in this line of research. So here is a picture of some ectopic tumors that have STAT3 driving luciferase. And so we want to be able to detect the amount of STAT3 activity using the luciferase in the mouse tumor, and then if we can modulate the STAT3 pathway, we should be able to see that luciferase signal go down. This is a little bit different system than you usually see because it's actually STAT3 driving the luciferase expression. So as long as there's signal, that means that STAT3 is on in that tumor. So you can see that STAT3 is highly active in these pancreatic cancer tumors when you put them on the flank of a mouse. Okay, so we are going to use a couple of tools to investigate APE's regulation of STAT3 DNA binding. One of those is the C65 mutant, and basically we have mutated the cysteine at position 65, which is the redox center of APE Ref1, and it does not affect the DNA repair activity. So this is a redox-dead mutant that still possesses full DNA repair activity. And then we also have a small molecule inhibitor called E3330 that we can use. This first slide just shows that we were able to reproduce the data from that cancer research paper showing that STAT3 DNA binding is redox sensitive, and we used EMSA assays to do that, or electrophoretic mobility shift assays. And you can see here when you stimulate the pancreatic cancer cells with IL-6, you indeed get more STAT3 binding to the DNA. here are two, a reducing agent and an oxidizing agent. You can see as you add more reducing agent, you get more STAT3 binding, showing that it is under redox control. And when you add the oxidizing reagent to that reaction, then you now negate the binding of STAT3 to DNA. The next step we did was to actually take and add pure APE1 protein to that EMSA assay. So again, we're looking at the ability of STAT3 to bind to DNA, and we add unreduced APE1 to the mix, and you don't see much more over just normal binding. Once you add the reduced APE1 to the cell lysate, now you can see that STAT3 can bind better to DNA. So we do know that when APE is present, that we are seeing an enhanced binding of STAT3 to the DNA. And this is just the DTT carryover that we use to reduce the APE1. So these beginning experiments told us that there was some sort of important reaction that was going on between STAT3 and APE1. So from here, we wanted to look at some of the overexpression of APE1 and look at proliferation. And so I just wanted to remind you of how we're doing that. And we're using the xCELLigence system from Acea. And one of the great things about this system is that we can monitor the cells in real time. We don't have to pick a 48-hour time point. Maybe what's actually important happens at 32 hours, and I never would have picked that time point. So that's one of the great things about this system. I can definitely show you that in the following slides. And as in the introduction, this basically measures the electrical impedance across these microelectrodes on the bottom of the E-plates. And another nice thing that they have now is E-plates where you can actually look through and see your cells. So you can monitor the health of your cells by your eye and your microscope, just like you would do in a 96 well plate if you're doing another proliferation-based assay. And so I can get quantitative information about the cell number, the viability, and the morphology. And you can see in this little diagram here, similar to the one that you saw earlier, after addition of a cytotoxic agent, you now have less cells, and so your cell index is going to change. Okay, so the first thing we did was we took pancreatic cancer cells and we overexpressed APE in those cells, and we overexpressed the Redox-dead mutant. So the C65A is the APE protein that does not have redox activity but is fully repair proficient. So this is using the xCELLigence system to just monitor the growth of the cells over time. And this is using, we did not do any kind of treatment other than just overexpression of the wild type protein, which you can see stimulates proliferation. And then here's our pcDNA vector control, growing fine over time. And then the C65 is not growing quite as well as that pcDNA, indicating that when the cells do not have fully proficient wild type APE1 in them, they are not growing as well as when we have more. So again, this is pointing toward APE1 being important in the proliferation of the tumor cells, especially. So then we took these same cells and we asked, okay, if we've got a whole bunch of extra APE in there, what does that do to the activity of STAT3? And so shown here is just two different clones, and we're looking at the pcDNA, the vector control, and then when you overexpress the APE protein, you see about a threefold stimulation of STAT3 activity. And then when you overexpress the C65 mutant, now that threefold stimulation is gone. So again, pointing to the fact that the APE's redox activity is having an effect on STAT3 DNA binding. The next thing we did was we wanted to take APE out of these tumor cells and see how that affects STAT3. We also wanted to show that this was actually at the DNA level and at the activity of STAT3. And so we did a Western blot on whole cell lysates showing that the phosphorylation of STAT3 is not changed when APE is knocked down, and the total amount of STAT3 is not changed. However, our knockdown worked. We just don't see changes in the activation of STAT3 at the level of phosphorylation. However, when we look at the basal levels or under situations of simulation, we see a dramatic reduction in the amount of STAT3 DNA binding if APE1 is not present. So now we wanted to again, show that APE and STAT3's interaction can be perturbed by blocking APE1 using our small molecule inhibitor, which we call E3330. So again, this is an EMSA assay where you're looking at STAT3 DNA binding sequence, and you can see that the basal levels of STAT3 binding are low. When you add APE to the mix, you get a stimulation of STAT3 binding to the DNA, and then as you increase the amount of E3330, the small molecule inhibitor, you see a decrease in STAT3 DNA binding, and that's quantitated here in the lower panel. We also did the STAT3 luciferase assay, looking at STAT3 activity under basal and IL-6 stimulated conditions, and the results were very similar. When you were inhibiting APE, you were also inhibiting STAT3 DNA binding. So the next step we wanted to do is see if we could dually target both STAT3 and APE using small molecule inhibitors. And so we used these three inhibitors that I mentioned, static, S3I, and the BP-1-102. And these were all selective small molecule inhibitors of STAT3. And this panel is very busy. Just suffice it to say that the STAT3 inhibitors are specific to STAT3. They did not affect STAT1 phosphorylation or STAT5, and that if you use enough of these compounds, they will inhibit pancreatic cancer cell growth. The S3I is not nearly as potent. As you can see, we have to go up pretty high with the doses to get inhibition with the single agent of STAT3 alone. The static is more potent, and you see it inhibits at much lower doses. However, the thing that was most striking to us is when we combined these two inhibitors together. So again, we're going back to that idea of synthetic lethality. Hitting STAT3 by itself is a good target. It does kill cells when you inhibit STAT3 in pancreatic cancer cells. And again, hitting APE1's redox activity also kills cells. But when you hit these two pathways at the same time, we see dramatic effects on proliferation. These were all done using the xCELLigence system, and these are 4 different pancreatic cancer cell lines. These 2 are well-established ones that you see in the literature quite a bit, and these are the low-passage patient-derived lines from Johns Hopkins University. The red in these panels is control, so those were treated with vehicle only. The blue is the STAT3 inhibitor alone. That's either S3I or the static. The pink, again, is another higher dose of the STAT3 inhibitor alone. And then E3330 alone is the green. So you can see we're using sub-lethal doses as single agents. However, the combination are the teal color and the purple. And you can see in each cell line using the S3I201 at 2 different doses, dramatic inhibition of proliferation. We really see they start out sort of okay, and then they really don't grow over time. And again, this starts to occur at 56 hours is where you see really nice separation. I never would have picked that time point if I was doing a time point based assay such as MTS or MTT. And each cell line is a little bit different. And a lot of times when you do different cell lines with an MTS-based assay, you would choose 72 hours or something like that and run all your cell lines at the same time. So that's another power of this technique. Now, in the patient lines, we do see that as single agents, the STAT3 inhibitors do work a little bit, but combined together, we see this dramatic effects. So this was exciting for us. We wanted to go ahead and make sure that this was we wanted to prove that they were dying via apoptosis. So we did some caspase-3 staining as well as an exon staining. This slide is just showing the caspase staining. And again, we're using 2 different pancreatic cancer cell lines. Here is the vehicle alone. Here is E3330 alone, and the STAT3 inhibitor alone, and then combination, and quantitated over here. So you can see dramatic effects, again, on the induction of caspase-3 when you combine the 2 inhibitors together. We saw similar results with the annexin 5 staining. We also went on to prove that these were synergistic using the Chou-Talalay method and generating combination index values. And you can see here these are strong synergies that when you combine STAT3 inhibition with APE inhibition. From there, we wanted to take the 2D model into a little bit closer to an in vivo model. So we went to using 3D cultures. And so these are, again, the low passage patient-derived cells. And this is using the STAT3 inhibitor alone. And so you can see we do see some cytotoxicity with STAT3 inhibitor alone. And then when you combine it with APE inhibition, you see dramatic cell death. So this is, again, just points to the fact that this combination is really working both in 2D as well as in 3D cultures. Okay, so the next couple of slides are going to be looking at some of the important cells that are supporting that tumor. And this is especially, as I mentioned earlier, in pancreatic cancer, because the extensive fibrosis that surrounds that tumor makes it very difficult to get the chemotherapy into the tumor. And so if we're just studying the tumor by itself, it's probably not a good representation of what's really going on in the cells. And so we're trying to really understand the disease and model it better by using the cells that are also helping that tumor to spread and helping that tumor to be protected from chemotherapeutic agents. And perhaps by doing that, we can do a better job of treating it. And so pictured here is the adenocarcinoma in yellowish red, and then here you can see all these cells that are supporting it and all the microenvironment that's important in making sure that that tumor is able to survive and ultimately metastasize. So this is an example of some of the 3D co-cultures that we're using. We've got these are have just been treated with a STAT3 inhibitor, and in this experiment, we actually labeled our fibroblasts with green. And so you can see in this panel, we've got tumor only, and then the fibroblasts only, and the co-culture. And we see, you can see the haze around the green, which is the cancer cells, and the tumor cells are unlabeled in this experiment because they're the low passage patient-derived cells, and so we want to keep them sort of in their purest form. And then here, when we add the STAT3 inhibitor, you really see the tumor cells are dying, but the stroma are still glowing green. So it looks like this particular inhibition of STAT3 looks to be more cytotoxic to the tumor cells than it does to the fibroblasts that are supporting that tumor. So now we've got to see if in co-culture, if we can get the tumor killed, even though it's in the presence of the fibroblasts that are helping it to grow. The other experiments that we've been doing is looking at how the tumor and the stroma are interacting using conditioned media. And so we use the cancer-associated fibroblasts, and we make conditioned media, take that media and put it onto the cells, and then we can monitor the proliferation using xCELLigence and qPCR. Basically, we can take the fibroblasts and knock APE down in those cells and then put them onto the tumor cells and see if they still grow. Or we can treat the fibroblasts with our redox inhibitor and show that when the fibroblasts have been treated, they no longer secrete the things that are important to stimulate proliferation. And I've been using xCELLigence to do that. So here, I want to draw your attention to what we're actually monitoring, which is tumor cell proliferation. But what I've added to the tumor cells is conditioned media from the black shows my control. So that was just media without any cells. And then in the red was conditioned media that was made from some stromal-derived fibroblasts called M2B1. And so you can see if you compare the black to the red that these cells, the media and growth factors produced by these cells definitely stimulate proliferation dramatically. And again, an important thing to notice here is that I'm able to go out to 5 days with the xCELLigence system, and I would not be able to do something like that in an MTS-based assay because I wouldn't be able to know my time point. So you can notice here that the curves really didn't start to separate until I was out at about 4 days. Then I also added, so now you want to compare the green is the control, no cells, with the drug, the APE redox inhibitor added to it. And so you can see that does not, no effect on proliferation of the tumor cells, very similar to control. And then, however, when you treated the stromal cells with the APE inhibitor, so APE's no longer signaling to STAT3, NF-kappa B, P53, all those proteins that are going to be driving proliferation type pathways, now you have shut down tumor cell proliferation just by inhibiting APE in the stroma. So these are some of the experiments that we're doing that really are nice to use the xCELLigence platform, because we don't have to choose an endpoint, a time point, when we don't have any idea what the correct time point would be. We can monitor over many days. And then the newest thing, and I don't have a lot of data yet with this because I've just started working with it, but the newest thing that I'm working on that I think is very clever is they have e-inserts. And what you can do there is similar to the conditioned media experiments, except instead of just pulling the media off the cell type that supports the tumor, you're actually having a co-culture. So pictured here is the e-insert. And in this e-insert, you can put just media, or you can put cells in there. So you can add the cancer-associated fibroblasts or stellate cells or other tumor cells, whatever kind of cell populations you want to look at, you can add that cell type in this upper chamber. And then in the lower, or the receiver plate, then you would add your tumor cells. So the cells do not come in contact, but the factors that they're secreting are then released into this media when I insert that plate into here, and then I can monitor tumor cell proliferation based on what these cells are secreting and making. And so an example of that is shown here, and you can see, again, this is monitoring tumor cell proliferation. I know nothing about how these cells are growing. All I know is the cells that are in my receiver plate. So you can see here, the red line is tumor cells that were not incubated with the cancer-associated fibroblasts. So, for instance, this would be in this well would be media only. And so you can see they grow, but they don't grow nearly as well as when you add a couple different concentrations of the CAFs. And so you can see both 1,500 CAFs in this well, and then over almost 4,000 CAFs in this well were able to nicely stimulate the proliferation of those tumor cells. So again, it's pointing to the fact that this interaction is critical. And now I can look at inhibition of STAT3. I can knock STAT3 down in the CAFs. I can knock APE down in the CAFs, and I can see how that affects tumor cell proliferation. Finally, the last little bit I wanted to show you is the migration assay. As Yama showed you earlier, it sort of looks like this. You put your cells in the upper chamber, and then you put your chemoattractant down below. We just serum starved our cells and then added FBS down here so that they would move. And after migration, we would see an increase in electrical impedance. And we also did the negative control for that experiment and did put no cells in top and got no signal, and as well as the negative control without chemoattractant, and the cells did not move. And all of that data is published in that PLOS One paper that that shows all this data in case you're interested in that. The negative controls we put as supplemental data. So here you can see these are my 3 agents that I've been using, 2 STAT3 inhibitors as well as an APE inhibitor, and these are just the single agents. And again, they do stop migration of cancer cells. So you can compare the red to the pink and the blue, and you can definitely see inhibition of migration. However, when you combine them together, so now you're hitting both STAT3 and APE1, you see this dramatic inhibition of migration. So now you're comparing, this pink is now the combination or the dual targeting, and you can see it works much better than either agent alone. And we went ahead and quantitated that in this panel. We also used the xCELLigence to monitor survival at this time point. So the way we got this graph was we just looked at the migration at 8 hours. And so we are able to export the data at 8 hours and then look over three to 5 different experiments. So I just got through showing you all this wonderful data about how if you dual target these two, you kill the cells. And so we were afraid that somebody might say, well, of course they're not migrating because they're dead. So we actually used one set. We used 2 cradles to look at migration and 1 cradle to look at proliferation in using the xCELLigence system. And actually at 8 hours, the dual targeting approach has not killed them yet. And as you saw in the earlier graphs, but it takes about 24 to 30 hours, maybe even 48 hours, to begin to see the dramatic effects on proliferation. So at 8 hours, these cells are alive, and you can see that here. And that's just graphed as the percent cell index of control. So we set our control at 100, and you can see that all of the treatments are still alive at 8 hours. So these cells are just not able to migrate. It's not that they're dead. So this is pretty exciting data. One, we've shown that APE1 can regulate STAT3 DNA binding, and that's another transcription factor that APE1 is regulating. And 2, we've shown if you can shut down APE1's redox activity and shut down STAT3's phosphorylation, that you can see some dramatic effects on cell survival. We also wanted to see what some downstream targets would look like, and so we just chose some well-established STAT3 targets such as cyclin D1, survivin, and BCL-XL, and this is just some of the data in one of the patient-derived cells. You can see that at doses that don't kill and don't do much for migration, we don't see much change in cyclin D1 expression. However, when you combine the two, now you're seeing cyclin D1 go drastically down. And here, the survivin levels are actually reduced quite dramatically with the STAT3 inhibitors, and even more so when you do the combination treatment. So again, this is pointing that we are hitting STAT3, and when you combine E3330 and the STAT3 inhibitors, you're hitting STAT3 in a more dramatic way. So we've also done this in mice. This is just showing that APE inhibition can slow down tumor growth as well as STAT3 inhibition. So now we're ready to do the combination, and hopefully that will work even better. So as a conclusion and future direction, we have been able to show that the dual targeting of APE and STAT3 is very deleterious to pancreatic cancer cells, and that we can still kill the cancer cells even in the presence of stroma. And we also are able to look at how APE and STAT3 are interacting in the stroma using some of these co-cultures and some of these conditioned media type experiments. And we believe if we can understand that better and we can understand the mechanism behind the inhibition of these 2 pathways, that we will be able to perhaps do a better job at treating pancreatic cancer patients someday. It's important to note that APE1 is not just regulating STAT3, but that it also is regulating HIF1 alpha and NF-kappa B. These are also very important pathways in pancreatic cancer and several other cancers. And so we believe that perhaps APE's regulation of STAT3 is one of the reasons that we see the dramatic synergy, but certainly not the only one. And perhaps you have to shut down multiple pathways and be upstream of several of these signaling pathways in order to get the dramatic synergy that we're seeing. I have to thank the people that helped me do all this stuff. That pictured here is my laboratory, and that's my postdoc, Yan Lin, and she's fantastic. She's worked really hard on this project. And Safi Shada was a fellow in the lab last summer, and now he's going to be working here full-time at IU, and so we're hoping to be able to interact more with him as he is in the clinic and can help keep the research clinically relevant, as well as several other collaborators that you can see here. So I just thank you very much for your attention and time, and I'm happy to answer any questions.